Imagine a life completely devoid of fear. Today, we delve into the extraordinary case of SM, the woman who couldn't be scared, and what her unique brain taught us about the human mind. Picture walking through a dark alley at 3 a.m. and feeling nothing. No caution, no unease. In 1994, researchers found a woman who experienced exactly that, SM. Researchers tried everything to frighten SM over the years, from deadly snakes to haunted houses, and they failed every single time. Why? The key lay in her extraordinarily rare genetic condition, Urbach-Wiethe disease. This disease had completely destroyed both of her amygdalae, the almond-shaped clusters deep in the brain that scientists believed controlled fear. What made SM remarkable was that she wasn't emotionless. She laughed, she loved, and she felt joy and sadness just like anyone else. Only fear was missing. Researchers at the University of Iowa saw an unprecedented opportunity to study a human life without fear. The experiments began immediately. They took SM to an exotic pet store, handing her snakes and tarantulas. Where most people recoil instinctively, SM was merely fascinated, wanting to touch them more. She remained calm through horror films and even a haunted house, where she poked one of the actors in the head, curious about the mask. Her heart rate barely changed. But then, researchers tried something radically different. They asked her to inhale concentrated carbon dioxide. Carbon dioxide is harmless, but triggers a primal suffocation panic in almost everyone. And for the first time, SM panicked. She gasped, begging them to stop. The woman who laughed at horror movies was terrified. This single experiment, decades into her fear-free life, completely rewrote neuroscience textbooks. Scientists realized fear isn't controlled by one region, but a complex network, revealing two entirely different fear systems working independently. The two systems separate how the brain processes external dangers versus internal bodily distress. The amygdala handles external threats, things you can see, like predators, enemies, or dangerous situations. Internal threats, like suffocation or poisoning, bypass the amygdala entirely. These signals go straight to the brainstem, our most ancient neural architecture. SM's brainstem was intact, explaining her panic during the CO2 test. But her story holds an even more profound lesson. Living without fear didn't make SM's life better. She'd been held at knife point and walked into dangerous situations because her brain couldn't generate those essential warning signals. Fear, it turns out, is not a flaw in human design. It's a feature. The unease you feel in a dark alley is millions of years of evolution keeping you alive. Today, SM's case informs modern treatments for PTSD, anxiety disorders, and phobias, teaching us that fear is not something to eliminate, but something to calibrate.